Hello and welcome to our YouTube channel. In today's video, we will be exploring the fascinating field of microbiology by delving into some basic terms used in this realm. So whether you are a student, a science enthusiast, or simply curious about microscopic world around us, this video is for you. First term is cell. The cell is the basic structural and functional unit of all living organisms. Cells are typically microscopic and contain genetic material and various organelles. DNA, deoxyribonucleic acid. DNA is a molecule that carries genetic information in most living organisms. It consists of two long chains twisted into double helix and contains instructions for developing, functioning, and reproducing all living organisms. RNA, ribonucleic acid. RNA is a molecule that plays a crucial role in protein synthesis. It is involved in transmitting genetic information from DNA to cell protein building machinery. Genome, the complete genetic material, that is DNA or RNA, in an organism or a cell. It includes all genes and non-coding regions of the genetic material. Aseptic technique, a set of procedures and practices to minimize the contamination of sterile materials or environments by microorganisms. It is commonly used in laboratory work and medical procedures. Agar. Agar is a gelatinous substance derived from seaweed commonly used as a solid medium to grow and culture microorganisms. Petri dish. A shallow, cylindrical, lidded dish made of a glass or plastic used to hold agar or other culture media for growing microorganisms. What is media? Media is a nutrient-rich substances or mixtures used to support the growth and cultivation of microorganisms in laboratory settings. Stain, a colored or fluorescent substance used to add contrast or highlight specific components of cells or tissues when viewed under a microscope. Gram stain. It is a staining technique that is used to differentiate bacteria into two major groups, that is gram positive and gram negative, based on their cell wall composition. Microscopy. The use of microscopes to visualize and study microorganisms, cells, and other small structures that are not visible to the naked eye. Culture media, a nutrient-rich substance or mixture that provides necessary nutrients and conditions for the growth and propagation of microorganisms in the laboratory setting. What is growth curve? It is a graphical representation that shows the growth of microorganisms over time, typically indicating different phases such as lag phase, exponential phase, stationary phase, and death phase. Next important term is mutation. A mutation is a permanent change in an organism's genetic material, that is DNA or RNA. Mutations can occur naturally or can be induced by various factors, and they can lead to genetic diversity and evolution. Virulence is also an important term. The degree of pathogenicity or ability of microorganism to cause disease. Virulence is often associated with the severity of the disease and symptoms. Antibiotic resistance. The ability of microorganisms, particularly bacteria, to withstand the effects of antibiotics, rendering them ineffective in killing or inhibiting the growth of bacteria. This occurs when bacteria mutate or acquire resistance genes, allowing them to survive and multiply even in the presence of antibiotics. Antibiotic resistance is significant concern in healthcare as it can lead to the failure of antibiotic treatments and the spread of drug-resistant infections. Thank you for watching this video. For more informative videos, please like the video and subscribe to this channel.